The Winogradsky column is a simple device for culturing a large diversity of microorganisms that consist of collecting wet mud from a pond in a bottle and mixing this bottle with carbon sources such as newspaper, egg yolks, and these both combined. After this, the bottles are incubed into sunlight for months, for a specific amount of time, until anaerobic or aerobic gradients are developed. These gradients produce specific microorganisms such as chromatium, chlorobium, etc. The objective of this project is to observe the samplings of the diverse microorganisms in our local community.
Background research. Our expectations were that parasites and insects would develop from the bottles, because many little animals such as frogs, snails, and worms live in the mud. So maybe we have collected some eggs from these little living things and the eggs will develop in our bottles. So while the time passes, the bottle will continue to have more and more insects. This hypothesis could be proved because the bottle is not completely sealed, so air can still pass through it, and the insects will feed from the nutrients of the soil. Our expectations were almost right because bacteria and other microorganisms are developed inside the bottles, but in what we were wrong was that no bugs or insects are present inside them.
In week 0, no change was observed in any of the columns. In week 1, the carbon column began to develop green small fungus. The sulfur column developed small white fungus. The soil at the carbon and sulfur column changed to a darker color, and the control column remained the same.
In week 2, in the carbon column, more small little black fungus appeared in the soil. In the control column, small little black fungus appeared. In the carbon and sulfur column, some parts of the soil turned white. And in the sulfur column, also some parts of the soil turned white and small black fungus began to appear.
In the week 3, in the control column, the soil turned to a darker color. In the carbon and sulfur, the soil became darker than the control column, and the white spots in the soil go to the top of the bottle. In the sulfur column, the soil became darker and also the white spots went to the top of the soil. And in the carbon soil, more black spots appeared at the bottom of the column. In week 4, in the sulfur column, the soil at the bottom of the column turned darker, and the white spots at the top of the bottle developed also black spots. In the carbon and sulfur column, the white spots in the soil developed black spots on it. In the control column, more small black fungus appeared at the soil, and at the end of the carbon soil, more black spots developed at the bottom, and the soil at the top of the column turned darker.